On April 20th, 2010, the Deepwater Horizon rig exploded in the Gulf of Mexico, killing 11 workers and setting off the largest accidental marine oil spill in the history of the petroleum industry.
We haven't had an oil spill of this magnitude at this depth and in this environment.
This was a systematic failure on the part of BP, the industry in general, and of the government regulatory regime.
Over the course of nearly 3 months, roughly 4.9 million barrels of South Louisiana crude gushed out of the Macondo well and into the Gulf of Mexico.
It was beyond my worst fears. It was something I hope I never see again.
The Macondo wellhead was located at 5,000 feet beneath the water's surface, discharging large amounts of oil and gas under enormous pressure.
Most other spills are associated with a surface breach from a tanker. The oil sits on top of the water. This was a completely different situation. This was the first deep water blowout.
2.9 million gallons of the chemical dispersant Corexit were applied at the surface and near the wellhead to break up the oil and keep it from soiling sensitive marshes.
Dispersants were a novel concept in terms of using them subsurface. There's a great amount of angst, not only in the public, but among the federal agencies. What are the limits of this? Is this a good thing or a bad thing?
I don't think anybody knows what the answer to that is. I understand why the heads of agencies made the decision to use a Corexit. They were trying to avert a worsening condition on the seashore in the sensitive nursery areas and the estuaries. By making that decision, they made the conditions worse offshore and at depth. It was a no-win situation.
Trying to understand the balance of those pluses and minuses in the long term is clearly something that we have to find out.
Opinions on the fate of the oil vary widely. It is estimated that anywhere between 13 to 60% of the oil remains in the Gulf of Mexico, potentially affecting ecosystem health for years to come. What will the long-term impacts be? How long until the true extent of this disaster will be known?
Major funding for this program was provided by the Batchelor Foundation, encouraging people to preserve and protect America's underwater resources.
The catastrophic oil spill in the Gulf of Mexico may no longer make headline news. But the response to the disaster is far from over.
Here we're on station in position.
Scientists from the government, academia, and independent research institutions continue to study the impact of the spill. Among them is the faculty of the College of Marine Science at the University of South Florida, who became involved early on.
We had research vessels sitting at the dock. We had personnel that had expertise in various areas related to the spill. And so we felt that we needed to react.
The USF College of Marine Science, located in St. Petersburg, Florida, is the home base for the Florida Institute of Oceanography's research vessel, Weatherbird 2.
We were one of the first vessels that were directly in the oil spill, and we worked closely with the federal government and with the state. And we did it for several reasons, because we felt like it was our responsibility. We are a public institution. We are here to serve the citizens of Florida, and there was tremendous concern about what this will do to our economy, to our tourism, which has been a big issue.
Prior to the Weatherbird's first research cruise in May of 2010, a group of local researchers and faculty members at the College of Marine Science gathered to come up with a strategy.
We had a long discussion about what would be the optimum sampling plan.
We had a couple of reasons for expecting there to be a subsurface layer of oil. One is some experimentation that was done that indicated the majority of the oil released at depth, at tremendous pressure, will not make it to the surface. The pure physics of having very small droplets of oil, which form naturally, but they're also created intentionally by dispersants such as Corexit. Those oil droplets have a very hard time fighting against the resistance of the water on their way up, and they end up being stationary.
The USF scientists also knew that shortly after the spill, a different group of researchers working from the research vessel Pelican had discovered an undersea hydrocarbon plume to the southwest of the Deepwater Horizon drill site.
And that clicked for us, especially with our circulation models that suggested both a trajectory to the southwest but also to the northeast in the direction of the continental slope and shelf of Florida.
So that narrowed things down very much to the point where we could look at the charts and pick a place and say this is likely to have oil at it. We also picked areas that we thought would not have oil for comparison.
We wanted to cover areas that were of more intrinsic importance to the state of Florida, as well as to cover regions that other investigators weren't covering.
So we had a number of stations selected at different distances from the wellhead at different depths that were designed to intercept any unseen oil moving toward the state.
Once at sea, it didn't take the researchers very long to find subsurface oil.
Finding it, we thought might be a needle in a haystack, but we found the oil within a few days.
It was a thick plume. We were able to recognize two components. One was at 400 meters, that spanned about 100 feet thick, and the other one was located between 1,000 and 1,200 meters water depth, which was essentially the bigger plume, and that extended for upwards of 600 feet. We were able to see these features in the subsurface simply using a fish finder.
Once back in the lab, Dr. Hollander and his team ran chemical tests to confirm the plume was made up of oil. Next, they matched or fingerprinted the plume to the oil released by the ill-fated Macondo wellhead. And the research didn't end there. Since May of 2010, the scientists have made repeated trips to the area. While at sea, experts use a number of instruments to collect data that might provide clues on the environmental impacts of the oil spill. Water samples are collected using a device called a Rosette sampler.
The Rosette sampler has various bottles that we can trigger at different depths. And the way we decide what depths to trigger at is based on sensors which are attached to the Rosette sampler. So there are sensors for fluorescence, temperature, depth, and also light scatter. And the light scatter is a crude indication of where there might be oil droplets.
The data from the sensors is retrieved back through an electronic cable so that on the ship we can see the data in real time.
Bottle 4. That's at 100 meters.
The scientists collect water samples at standard depths, as well as in areas where they observe some unusual features.
All right, could you bring her up to 800, 800?
The water samples collected are then used for various analyses. One technique can quickly detect the presence of hydrocarbons in the water.
The technique is based on the fluorescence properties of some chemical compounds. What we do is we shine light of different energies on the sample, and we collect all the emission at different wavelengths. This gives us a three-dimensional picture of the total fluorescence of the water in our sample.
So at this characteristic level here, which is in the ultraviolet range, that's still below the detection level of the human eye, that's a telltale sign of a petroleum signature in the water.
Samples that test positive for hydrocarbon presence are analyzed further and fingerprinted to see if they are from the Deepwater Horizon spill site. Other USF researchers along on the cruises analyze water samples from the same sites to see if they detect toxicity. To do so, they use 2 different tests or assays.
So for this assay, we're using a living organism, a type of phytoplankton called dinoflagellate, as an indicator whether or not there's something harmful in the water, something that will negatively affect its metabolism. So these dinoflagellates bioluminesce, or they glow, and produce light as part of their natural metabolism. And when they are stressed, they will produce a lower level of light. If there is a decreased light emitted, there's something present in the environment that is making them unhappy, whether it be hydrocarbons or something else. Essentially, we're looking for a correlation between the toxicity data we come up with and data collected by scientists that shows the presence of hydrocarbons. So if we can correlate toxicity with presence of hydrocarbons, that would provide evidence for a biological effect in the environment.
The second test or assay uses a bacterium instead of phytoplankton to search for toxicity.
No one organism reacts to all the toxicants. What we found was that the bacterial assay was more sensitive to oil. The dinoflagellate assay was more sensitive to the core exit.
These tests show if there is an acute toxicity present, that could have an immediate impact on the ecosystem. Scientists are also very concerned about the impact that low-level, long-term exposure could have on marine species and ecosystem health.
The chronic toxicity, the subacute toxicity, we haven't measured, and we don't know what the impact of that's going to be on the food chain, on fisheries, and the health of the Gulf of Mexico.
The analogy is you can stick your nose into a bucket of paint, and you can sniff for a minute and get a headache, a raging headache, or you can paint your room and sleep in the room overnight and wake up with the same headache. So you can have a lower concentration in the surrounding environment, but it be exposed to it for a longer period of time, giving the same toxic response.
Another toxicity assay looks at the potential of toxins to cause DNA mutations. Of the water samples collected in August 2010, about half were genetically toxic. Tests conducted as recently as February 2011 continued to find genetically toxic samples.
And that means mutagenic, or the capability to damage DNA and cause mutations, and mutations can lead to cancer and other bad effects in marine life. And the other thing about DNA damaging agents is that it becomes a heritable trait. If there's a mutation that's caused, it can be permanently changed in that particular breed stock or organism. So we're very concerned about those types of toxicities. One of the most susceptible to genetic toxic materials are larvae. And these are organisms that are just rapidly growing, rapidly dividing, synthesizing a lot of DNA. Now the problem is that the larvae won't turn up to be adult fish for maybe 3 or 4 years, so we might not really see the genetic impact for a couple years, and this might manifest itself in any number of features.
Another group of USF scientists is focusing their research on the impact the spill is having on the bottom of the food chain.
The part of the food web that our group is targeting is the lower end of the food web, which includes the small microscopic plants and animals.
Those microscopic plants and animals are known as plankton,
which means small organisms that drift with current. Phytoplankton are the microscopic plants, so they're unicellular plant life. The zooplankton are the small microscopic animals. They range in size from microscopic, which are the same size as the plants, to individuals like small fish larvae, for example, that you can see with the naked eye. And we do a variety of different analyses to determine what the composition is of the phytoplankton and microzooplankton, how many are there, and how that changes through time. And so what we're trying to observe is any differences from a normal natural variability.
Observing such potential changes is a bit of a challenge for scientists.
The Gulf of Mexico has been understudied, so we didn't have really good baselines for comparison.
It's a very complicated system. I always say the Gulf of Mexico used to be the Rodney Dangerfield of oceans. It never got any respect.
We need a lot more attention paid to establishing these baselines of all types so we know what normal is.
If we had had years of data to this point where we knew what the natural variability was over space and between week to week, month to month, year to year, we would be able to determine much more accurately what the effects of the BP oil spill were.
Despite some of the uncertainties, scientists say they have observed a change in the abundance and health of plankton, and they expect to see continued changes over the next few years. An increased mortality of plankton could have serious implications for the entire food web.
We all fear what we call regime shifts. When you have a large-scale change in the dominant animals of your ecosystem that's very stubborn, once it switches over to this new order, it doesn't go back. And so that would affect fishermen and cultures all around the Gulf of Mexico if all of a sudden a certain species of animal that's been harvested for generations has been replaced by another one.
Very concerned about things like Atlantic bluefin tuna, which a large fraction of the stock spawns in the area where there was oil in the water. And so that's a depleted stock as it is, and so any factor that might reduce the number of animals that might recruit to the population is of concern. One of the things that's kind of a truism of these big environmental events like oil spills is that we need to expect the unexpected, that there may in fact be these sublethal effects that will only reveal themselves over time. And we certainly saw that with Exxon Valdez, and it's very likely we will see that here.
In the case of the Exxon Valdez, 4 years went by and the local herring stock was reopened for fishing, and there was immediate collapse in that fishery. And that stock has not recovered since. Decades have passed.
To determine the impact the Gulf oil spill is having on fish stocks, USF researchers are using an innovative method.
We're looking at the chemistry of otoliths. Otoliths are ear bones that occur in the fish's head. They're laid down in layers like an onion or tree rings, and they incorporate the chemistry of the water around them. So you end up with a permanent record of the history of the water that the fish was swimming in. So to get that record out of the otolith, we cut very thin sections out of the middle of the otolith, and then we polish those thin sections, mount them on a slide, bring them to this machine, and it has a state-of-the-art laser that is used to sample the different types of elements that are recorded in the rings. So if this fish were exposed to the oil spill, you would see unique elements showing up in this transect. So what we're looking for is, number one, whether or not the fish was exposed, and number two, whether or not the fish experienced slower growth rate after it was exposed. Slower growth rate in fish generally translates into higher death rates. So growth rate is not a trivial thing. If we see that there was a reduction in growth rate associated with exposure to the oil, then that means something negative is likely to be influencing the population. We have a lot of samples of important recreational and commercial species, including red snapper. We have large predators, we have small fish from the deep ocean that people generally never see, even if you spend your entire life on the Gulf, things that we've trawled up from a very deep part of the Gulf of Mexico. So we have a comprehensive spectrum of species that we'll be looking at.
Studies suggest that the oil in the water column is diluting, and some of its components are being broken down by microbes. As more time passes, experts say it is becoming more difficult to find traces of oil in their water samples.
The one place that still seems to be at potential risk is the sedimentary environment, both in the shallow but also in the deep marine system where the plume was in that region.
To study the sediments, experts collect samples with a device called a multicore.
There's a central column that has 8 separate cores that are set up that when the device hits the sediment floor, that main column impales itself into the sedimentary system, closing then these vacuum-type seals, and then the activated sample pulls up, a bottom arm swings underneath the core and traps the core material inside and brings it to the surface.
A depth is 1002 meters.
Sediment samples are cut into thin slices and spun in a centrifuge to separate the water from the mud. This water, called pore water, is then analyzed for toxicity and the presence of hydrocarbons.
This is a shape and a region that's characteristic of petroleum. We do need to do further analysis to say, oh, this is exactly one compound or another compound, and I'm in the process of doing that right now. We've got some PAH standards. PAH stands for polycyclic aromatic hydrocarbons, and those are some of the most toxic compounds that are in the petroleum mixture. So what I'll do is I'll analyze those known standards, and I'll be able to compare those to what I've seen in the natural environment.
PAHs are not only some of the most toxic components of oil, but they also take a long time to break down. To date, USF scientists have found that pore water samples from the sediment cores have tested positive for toxicity, as well as the presence of hydrocarbons. Many of those samples were taken in an area known as the DeSoto Canyon.
We knew from prior history that that was a very important area for fisheries. Canyons can also focus the downward flux of material as well. So we anticipated that that might be an area where we would also see an impact on the seafloor.
The sedimentation rates in those areas of the DeSoto Canyon is about 0.3 millimeters per year. 0.3. So in 3 years you get 1 millimeter. That's under normal conditions. What we're recognizing is that the sedimentation rate has increased by about an order of magnitude. So now we're looking at 3 to 10 millimeters per year. So a significant increase in sedimentation rate, and the question is why? And so there's a number of hypotheses. The most prevalent one is the blizzard hypothesis, and that is that the oil droplets, maybe with the addition of the dispersants, has lent itself to a very sticky type material that aggregates other biological material, and it ballasts it so that it eventually sinks. And if there's enough of this, you can actually smother the habitat below.
Another theory on how the sediments and marine life on the bottom of the sea may have been impacted is known as the bathtub ring hypothesis.
The plume, as it impinges on the continental slope, would leave a bathtub ring of petroleum products.
In some areas, the small creatures that live on the bottom of the ocean appear impaired or dead, and scientists say more research needs to be done to understand what is happening to the ecosystem. Past oil spills, such as the Exxon Valdez in Alaska and the Itztock spill in the southern Gulf of Mexico, have shown that toxic oil can persist in the environment for decades.
The Gulf of Mexico is one of the 3 largest oil provenances in the world. Oil drilling, oil and gas exploration in the Gulf of Mexico is here to stay.
We really need to have this well thought out in terms of different scenarios, and that can only occur if you've got research that's relevant to the different problems that might occur. We need to actually make some serious investments in this so that the next time around, and there will be a next time, that we're not sort of offering platitudes as opposed to actually having hard quantitative information available.
Many questions remain about the long-term impacts of this oil spill. Researchers from the University of South Florida and many other institutions are doing what they can to better understand the full implications of this disaster.
A lot of people are working very hard doing methodical tasks, and often it's not until later that you realize the significance of what you found. It'll take many years before we understand the impact, and it'll take even longer before we can put the impact into context. Putting the spill into context is definitely something that will take decades.
Major funding for this program was provided by the Batchelor Foundation, encouraging people to preserve and protect America's underwater resources.